Hello students, this is J.K. Sharma Bioclasses and we are continuing the chapter 5, the fundamental unit of life. In this section, we will study about cell organelles and cell inclusions. In the previous section, we had already studied the structural organization of cell like plasma membrane, nucleus and cytoplasm. In this section, we will study about cell inclusion and cell organelles. Today's topic is cell organelles and cell inclusions. In cytoplasm, students, we see many structures. Some are membrane bound structures and some are not membrane bound. So those structures which are membrane bound, they are known as cell organelles, and the structures which are not bounded by membrane are called cell inclusions. The membrane bound structures present in the cytoplasm are called as cell organelles like mitochondria, chloroplast, nucleus, endoplasmic reticulum, etc. And the structures which are not bounded by membrane are called as cell inclusions like ribosomes, centrioles, etc. So we'll study them one by one. You can see here this is animal cells and this is plant cells. In both the cells, their cytoplasm have cell organelles and cell endoplasmic reticulum. Can you tell me whether it is cell organelle or a cell inclusion? Yes, this is a cell organelle because this is a membrane bound structure and this consists of an irregular network of channels. You can see in this picture. So this is like a network and these are membrane bound and all these channels are interconnected with each other. They can only be seen with the help of microscope. And which microscope? Electron microscope only, because this is very microscopic structure. There can be two types of endoplasmic reticulum. First one is RER, that stands for rough endoplasmic reticulum. ACR, that stands for smooth endoplasmic reticulum. RER is characterized by the presence of ribosomes on their surface. If ribosomes are attached on their surface, it is present on their surface, then it will be called as rough endoplasmic reticulum. You can see here tiny ribosomes are present, and if ribosomes are present on the surface, then it is called to be rough endoplasmic reticulum, and is very important because ribosomes are the sites of protein synthesis. So this endoplasmic reticulum helps in protein synthesis because of the presence of ribosomes on their surface. Now second one is ACR. ACR means smooth endoplasmic reticulum. Simply, if ribosomes are there, then it is rough. If ribosomes are not present, then it will be called as smooth endoplasmic reticulum. On these endoplasmic reticulums, ribosomes are not present, so it is called smooth endoplasmic reticulum. And this is important for lipid synthesis. Lipid means fat synthesis. And we have studied plasma membrane is made up of phospholipid and protein, so it helps in membrane synthesis. You can see here the large view of endoplasmic reticulum. These are smooth endoplasmic reticulum and this is rough endoplasmic reticulum. Can you tell me why is it called to be rough endoplasmic reticulum? Because it has the presence of ribosomes on their surface. What are the functions of endoplasmic reticulum? Ribosomes present are the sites of protein synthesis. We have already discussed ribosomes are the sites of protein synthesis, so it helps in protein synthesis also. Smooth endoplasmic reticulum helps in manufacture of fat molecules, or we also call it lipid molecule, that helps in formation of lipids and synthesis of membrane. It helps in intracellular transport of the substance like proteins. It helps in the transport of proteins between the cells, inside the cells. It helps in the synthesis of steroid hormone. Steroid hormone like Many hormones are there: progesterone, testosterone, estrogen. These are all steroid hormone, and these steroid hormones is synthesized by the help of endoplasmic reticulum, and it acts as skeleton system of the cell because it forms a network like structure, so it provides rigidity to the cell and it becomes a skeletal system of the another cells. Very important functions of smooth endoplasmic reticulum, that is membrane biogenesis or membrane synthesis. Smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipid molecules, and these are important for cell functions. And some of these proteins and lipids help in the building of cell membrane. We have already discussed cell membranes are made up of phospholipid and protein. So these lipid and the protein, these are the building block of the cell membrane, and this process of formation of cell membrane by the help of smooth endoplasmic reticulum is called membrane biogenesis. Another important cell organelle in the cytoplasm is Golgi apparatus or Golgi body. This is also a membrane bound, so it is cell organelle, and this was discovered by Camillo Golgi, so named Golgi apparatus or Golgi bodies. These are membrane bound or single layered membrane bound structure that occurs in the form of tubules, vesicles and cisternae. You can see at the top of the Golgi bodies there is round structures at the head. This is called cisternae, and some are round structures, oval structures. This is called secretory vesicles and tubules, and some are this you can see like tubules. So it can be in the form of tubules, vesicles, this lobe-like structures at the head, and another oval shaped structures, round shaped structures. They are called as vesicles. They are usually seen aggregated towards the nucleus. You can see this is the nucleus here, and this is Golgi apparatus. एक ही जगह पे सभी ऐसा लगेंगे कि जमा हो गए हैं, इस तरह से ये दिखाई देते हैं. In plants they are unorganized, और plants में ये unorganized होते हैं, और they are not interconnected, they are unconnected channels like structures, and these structures is called dictyosomes. The structure is called dictyosome. So Golgi apparatus in plant, it's called what? Dictyosomes. You see the larger view of Golgi apparatus. We can see here these are vesicle form of, these are tubular form of, and this at the head you can see these are cisternae. Functions of Golgi bodies. Golgi bodies help in the synthesis of some polysaccharides from simple sugar. Polysaccharides like glycogen, starch, chitin, cellulose, etc. And simple sugars are like glucose, fructose and galactose. It helps in the formation of glycoprotein. It is involved in the transformation. Transformation means the change of one form into another form. Like one biomolecule will transform into another biomolecules, and one structure will transform into another structures. Primary lysosomes are formed from Golgi bodies by budding. So this is one of the examples of transformation. And the Golgi bodies are responsible for secretion and transports in the cell. Lysosome. Lysosome is a cell organelle present in the cytoplasm of the cell. It is a membrane bound structure. It is bounded by a single membrane. This lysosome contains hydrolytic enzymes or digestive enzymes. Lysosomes are also called as suicidal bag of the cell. आत्महत्या की थैली. क्यों? Because during the disturbance in the cellular metabolism, when the biological process in the cell is not going properly, is not if the cell is not active, and if cell gets some injury or cell gets damaged, in that situation, what happens? In that condition, in that situation, this lysosome may burst, and all the enzymes of this lysosome comes out, and it digests their own cells, and along with the cell it also dies. So what does it do? It commits its suicide, and that is why it is called suicidal bag of the cell. सेल के साथ ये भी खुद भी मर जाता है. Enzyme present in the lysosomes are made by RER. RER we have already studied, rough endoplasmic reticulum. They also destroy the foreign materials which enter the cell, and it also protects the cell from external particles of foreign materials, and these foreign materials may harm the cell, so it protects the cell, it digests the foreign materials with its enzyme. So in this way it is protecting the cells also from external bodies or foreign bodies. Here we can see this, the larger view of the ribosome. Ribosome, you can see this picture. So these are oval shaped small structures, this vesicle like structures, and these vesicles are formed from Golgi bodies, and this contains hydrolytic enzymes, and these hydrolytic enzymes, as we have seen, because of this enzyme it is called suicidal bag of the cell, or it protects the cell also from the foreign bodies. Functions of lysosome. Lysosome helps in digestion of food within the cell, and the digestion within the cell is called intracellular digestion. Like in amoeba we have seen, there is pinocytosis or phagocytosis takes place in the cell. Sometimes cells takes engulf solid material, sometimes it takes in liquid material into it, and this is digested with the help of the enzymes of lysosome. Second one, it protects the body from bacteria and other harmful organisms because of the presence of its enzymes. It's a hydrolytic enzyme, it's lytic enzymes. They help in removal of old worn out cell organelles, because this enzyme can digest the damaged cell organelles and damaged part of the cell by digesting them. Mitochondria. Mitochondria is one of the important cell organelles in the cell, present in the cytoplasm of the cell, and these are small bodies, and they are present in the cell in huge number, in large numbers. Each mitochondrion consists of two membranes, outer membrane and inner membrane. You can see here, this is outer membrane and this is inner membrane. You will see that inner membrane has got some inward folding. अंदर की तरफ कुछ foldings दिखाई दे रहे होंगे. So these foldings of inner membrane is called crista, and crista plays very important role in respiration. Crista lie in the ground structures, and this ground structures, this fluid part of the mitochondrion is called matrix. This blue structure we can see, this is matrix, and this matrix contains a large number of required oxidative enzymes which helps in aerobic respiration and to release energy, to form energy in the form of ATP. That is why mitochondrions are also called as powerhouse of the cell, because it contains oxidative enzymes and they can generate energy in the form of ATP. That is why it is also called as powerhouse of the cell. They have their own DNA and ribosomes. Important, the mitochondria, semi-autonomous body. They are self-reliant, isn't it? Let us see function of mitochondria. Already we have discussed. Mitochondria have enzyme necessary for the oxidation of glucose, thus releasing energy in the form of ATP, and they are therefore called as powerhouse of the cell. We have already discussed. Plastid. Plastid is only present in plant cell. It is not present in animal cells. Plastids is the characteristic features of plant cells only. It is of two types, chromoplast and leucoplast. Chrome means colored, so this is colored plastic. Leucoplast, leuco means white or colorless, so this plastic is colorless or white plastic. This is a lamellated structure which is bounded by double membrane. As mitochondria was also double membranous structure, this is also two membranous structure, outer membrane and inner membrane. You can see inner membrane and outer membrane. Plastid containing the pigment chlorophyll, and this chlorophyll is a green pigment. The plastid which contains chlorophylls are called chloroplast. And chloroplast is very important for photosynthesis. Photosynthesis is the process by which a green plant make their food in the form of starch in the presence of sunlight with the help of water and carbon dioxide. Leucoplast stores starch, food, protein granules. So these are the food materials. So leucoplast stores food reserves. Plastids show two distinct regions, grana and stroma. We can see here this part, जो stack of coin-like structure है, this is called granum, and the matrix inside the chloroplast, the fluid part of the chloroplast is called stroma. Plastids have their own DNA and ribosomes like mitochondria. So plastids can also be called as autonomous bodies. Autonomous bodies. They are self-reliant. They can divide on its own and they can synthesize protein also. So here the important part is this chloroplast contains chlorophyll, a green pigment. Leucoplast stores food reserve materials, and plastids have two parts, grana and stroma. Grana is a stack of thylakoids and stroma is the matrix, the fluid part of the chloroplast or plastid. Let's see some different kinds of plastids. Proplastids. Proplastids. This plastid is undifferentiated and this is colorless. Another plastid is etioplast. Etioplast does not have any chlorophyll, so and this is not greenish. Other than greenish, it can be any other color, but this won't be green in color. Chloroplast will have green color chlorophyll. This is a green pigment and this helps in photosynthesis. These are present in leaves and stems. Amyloplast, as we have seen in leucoplast, it stores starch. It's called amyloplast and it is colorless. This is one of the leucoplast and this is present in roots, seeds and fruits. Chromoplast. This chromoplast is colored plastid. It may be red and yellow colored. These are carotenoid. Carotenoids are yellow in color, and xanthophyll, one more chromoplast is there, and this is present in the flower or fruits, which make them look yellow. So petals and fruits, these chromoplasts are present in petals of the flowers and fruits. It gives a different color to the petals and fruits. Let's see the structure of chloroplast. Chloroplast is green plastids, and the plastids containing green pigments are called chloroplast. And this chloroplast is made up of two membrane, inner membrane and outer membrane. And it has two very important parts, granum and stroma. Each coin-like structure is called thylakoid. And when there is a stack of thylakoid, this stack of thylakoid is called granum. And there is fluid part in the chloroplast, and this fluid part, a matrix of the chloroplast, is called stroma. And this is the site for dark reaction, and this granum is the site for light reaction for the photosynthesis. and there is intermembrane space. Intermembrane space means the space between two membranes, and this chloroplast is very important for photosynthesis because it contains a green pigment chlorophyll, and chlorophyll is very much required for photosynthesis. Functions of plastids. First function, chloroplast is also called the kitchen of the cell. Why is it called kitchen of the cell? Because food is synthesized by the process of photosynthesis, and for photosynthesis green pigment is required, and green pigment is present in chloroplast. And what is the name of that green pigment? Yes, chlorophyll. Chromoplast gives various color to different parts of the plant, petals of flower, fruits, etc. Chromoplast is colored plastids like xanthophyll, carotenoid, etioplast and all that. Leucoplast stores food. It is colorless plastid, but it stores food. It is present in the starchy food. It is present in root and some other parts which stores food. Vacuole. Before we start vacuole, I would like to ask you a question. Name two cell organelles which contain DNA and ribosome. Yes, mitochondria and plastids. That's why they are also called as semi-autonomous structure of the cell. Now vacuole. Vacuoles are sac-like structures, and this is meant for the storage of solid and liquid material. It stores a solid or liquid material. If it stores solid material like amoeba stores solid material like food, and that is why this vacuole in amoeba is called food vacuole. Vacuoles are small sized in animal cells, but it is large size in plant cell. It occupies most of the part of the cell in plant cell. In plant cell, vacuoles are full of cell sap that contains amino acids, water, protein, organic acids, sugar, etc. And this cell sap provides turgidity and rigidity to the cell. It gives the cell a strength and a proper shape. The outer membrane of vacuole is called tonoplast. And what is the function? It is osmoregulatory in function. It regulates the osmotic concentration of the cell sap. We can see the large picture of vacuole, and in plant cell it is bigger in size. In animal cell it is very small. Vacuole's outer membrane of the vacuole is called tonoplast, and it helps in osmoregulation as well as in excretion of waste material from the cell. Thank you. In the next section we will study about tissue. Stay home, stay safe, stay healthy. Thank you.